be talking about Stachybotrys. And before we get into too much depth, I just wanted to do a quick disclaimer that a majority of the information that I will be using throughout my slides is specifically for a species called S. chartarum. Today we will also be learning its virulence factors and exploring something called mycotoxins, which is one of the most interesting things about it, especially how these toxins contribute to disease and the effects that they have on human health and ways to avoid them. To get us started, let's go over the morphology. For S. chartarum, it is actually a filamentous fungi, kind of like most molds, where they are composed of these thin structures called hyphae. You can see it at the bottom left over here. Since it is a fungus, it is eukaryotic, and it also has chitin, which is the main component of cell walls. One other thing we'll be going over later is orgesterol. We did talk about this in our first chapter together. It'll be way more important later on. In these hyphae, it's split up into two major parts. There are the phylidae, which are these little nubs at the end. They are flask-shaped. You could see them in our laboratory right now. They're kind of like test tubes, like more triangular. Similarly, they actually produce condia as spores. Because they are spores, they're used for asexual reproduction, genetic persistence, and survival. And one of the biggest reasons that it is called black mold is because it is highly melanated, kind of like how our skin can be melanated. So it picks up a natural, like green hue-ish gray or brown conifers, which are the little spores. And just to help visualize everything, I did add the taxonomy and classification. For one, it is a eukaryote, meaning that it does have a nucleus. It is in the kingdom of fungi, more specifically the division of Axomycota, meaning the kingdom that grows fruiting bodies, aka the actual visual looking mushrooms that you could see outside, more specifically a sac mushroom, which is pretty interesting. I wouldn't get into it right now though. The family is Stachybotryaceae, and the genus of Stachybotrys and chartarum. Stachybotrys chartarum is actually found worldwide. You could commonly find them growing in buildings, but they are a pretty versatile mold. All they need is a decent amount of moisture, but they could grow in almost any sort of pH range, going from 3.0, meaning highly acidic, to 9.8, meaning highly basic. As for their temperature range, they optimally grow about 68 to 80 degrees Fahrenheit, meaning that they are mesophilic, but there have been some cases where they have been found to grow at 35 degrees Fahrenheit. One of the most interesting that I found is a deep ocean variant species of chartarum that is found growing in corals. And as for the risk factors, not everyone is affected equally. One of the most important infections that have been caught on record is through animals. Most of the time when the mold is found growing on, let's say, the hay or straw of farm animals and been digested, it led to very severe effects. Most importantly, heart arrhythmia, meaning that your heartbeat doesn't stabilize, it beats every so often, skips a lot. There's been necrosis where your live skin cells die off. And there have been cases that I'll talk about soon in children and pregnant women where it does affect the baby's development. And it is very severe if you are immunocompromised, meaning that your autoimmune system does not fully work against the fungus. Some of the first ever recorded cases of S. chartarum is what is known as sick building syndrome, where people used to think houses or specific buildings were cursed because people that lived in them eventually started feeling sick, nauseous, queasy. To continue off of that, the fungus can be opportunistic. Not always. They're not known to make people sick actively. They are spread by guttation droplets, which means extra moisture from the fungus is excreted through the cells. And one of the biggest issues that people have while surrounded by black mold is mycotoxins. It's a secondary metabolite, meaning that it is not needed or not essential for life. It's essentially a byproduct, and these are produced in response to the spore amount that is found produced. And now onto one of the most interesting bits of this fungus, the mycotoxins. For ease, I only brought up 3 of the 400 different recorded mycotoxins. There are tryptosines and atrinones. Those are very important, and they are also mutually exclusive with each other. So if you find another Stachybotrys, it could have either or, but not both. And just to keep it a little bit more brief, I only picked out the 3 most researched satrotoxins, which are in the tryptosine group. So for these satrotoxins, they actually bind to 60S ribosome subunits inside human cells, and just eukaryotic cells in general, which inhibits protein synthesis. Because of this, like I mentioned before, it led to mass death of livestock, like the pulmonary hemorrhages, the cardiac arrhythmia, plus it is found to do brain damage and liver damage to infants, meaning that they were born with gray skin, failing livers, and more importantly, overall, it inhibits protein, RNA, and DNA synthesis. Moving on to atrinones, they are actually antimicrobial properties that focus mainly on prokaryotes. They have not been studied as much as tryptosines, but they still are pretty dangerous. Even though we don't know the cause, they still cause severe DNA damage and cell death. And one of the coolest ones, in my opinion, is phenospirodrimanes. They are immunosuppressive, meaning that they weaken the immune system. And one of the first ever cases were not too long ago. It was an invasive species of Stachybotrys growing inside of a patient with acute leukemia, and it caused an infection. This was one of the first ever cases recorded. All the other cases have not been infectious. Thankfully, these mycotoxins are not as harmful as you think. A study earlier this year in 2026, they actually did find that 1 of 8 of the compounds produced by these mycotoxins could actually be used to lower gout. They specifically lower the IL-1B protein, which is a cytokine that normally promotes inflammation for specifically gout in this case. And they was also found to block the NF-κB inflammatory signaling pathways, which we did talk about as one of our most recent classes, which is basically a protein used in transcription and the production of cytokine. And it also lowered the ROS, which is the reactive oxygen species, which contributes to tissue damage. Some of the quickest ways to diagnosing yourself with the exposure to black mold is, of course, seeing the colonies. They cause some pretty nasty effects, like sneezing, coughing, sometimes even red eyes. A lot of the time it can get more severe, where you're basically asthmatic, you have a dry cough. Another quick, easy way to see if you might be affected by black mold is lessening when not exposed to it. So when you're out of the house for a while after feeling terrible, it could be a sign that you might have black mold. Most colonies can grow anywhere and usually are hidden away from us, so people exposed don't usually know that they are. So if there are any doubts, please find a professional to test for either the fungal spores or the mycotoxin itself. Similarly, there is Nosy, which is a really nice test. As for treatments, it's usually easier to kill the mold outside of the human body, be it bleach, removing the items. One of the most prominent researchers of black mold recommends either a sodium hypochlorite solution, hydrogen peroxide, or even isopropyl alcohol. And as for the spores themselves, they need to be killed off in an autoclave, which means that it has to hit at least 200 degrees Fahrenheit, or about 93 degrees Celsius, for about 15 to 20 minutes. And to end things off with an overview, Stachybotrys, specifically S. chartarum, are not pathogenic. Regardless, try to stay away from the areas that you might expect mold to grow, and Of course, the fungi produces secondary metabolites that have mycotoxins, which is the main cause for sickness. It is not deadly initially, but it can be dangerous to those who are immunodeficient, like young children, pregnant women,